Welcome to Science Breakthrough from Science Mission. I'm Sadashiva Pai and going to discuss a couple of breakthrough papers published in the week of May 19th. Let's start. First, we are going to discuss a paper published by Anna in Nature. Why this paper is so important. Metastatic carcinomas relapse within 6 months of drug treatment. This paper provides a possible mechanism for drug resistance, thereby treatment option for metastatic melanomas. First, let's look at the kinase pathways in cancer. BRAF inhibitor, that is BRAF inhibitor, blocks BRAF ERK pathway but develops resistance after about 6 months. There are alternative pathways that could lead to cell proliferation or tumorigenesis. Authors looked at these alternate pathways via MKK or MLK that act via P38 and JNK and identified the mechanism. Authors show that expression of constitutively active MLK124 resulted in activation of MKK4, MKK7 and JNK pathway similar to wild type, while empty vector and kinase domain dead protein did not activate. In addition, MEK and ERK pathways were also activated by MLKs. Authors also show that this activation requires kinase domain of MLKs. Constitutively active KD mutants for MLK3 and MLK4 also activated MEK ERK pathway, indicating a functional kinase domain is required for activation of the ERK pathway. Also, exogenous expression of other related kinases did not alter signaling through MEK ERK pathway, indicating specificity of the activation. Data is not shown here. Authors overexpressed MLK and treated the cells with RAF inhibitor L779450 or a MEK inhibitor U0126. MLK124 activated MEK ERK pathway in the presence of RAF inhibitor but not MEK inhibitor, indicating that MLK directly phosphorylates MEK. Next, MLK was induced in melanoma cells with tetracycline and treated with RAF inhibitor PAX4032, which is nothing but the drug used for cancer treatment. MEK ERK pathway is still induced. Similar result is also obtained by RAF siRNA introduction in a separate experiment. So MLK acts independent of RAF. Next, authors also show that MLK induced melanoma cells with tetracycline and treated with RAF inhibitor survived better than the ones that are not induced with tetracycline. MLK induced abrogation of cell death. All 4 MLKs abrogated the reduction in XIAP levels as well as cleavage of PARP and caspase 3 in the presence of BRAF inhibitor, suggesting that MLKs can mediate resistance to drug-induced apoptosis. Increased expression of different MLKs are also seen in patient tumors treated with RAF inhibitor, further confirming role of MLKs in tumor progression. Authors came up with 2 models based on their animal study to demonstrate mode of resistance to RAF inhibitor. One, acquired resistance, wherein treatment of RAF inhibitor results in upregulation of MLKs, which in turn directly activate MEK ERK pathway. The second one is the de novo resistance, wherein gain of function of MLK activates MEK ERK both in the absence and presence of RAF inhibitor. These studies conclusively prove a role for MLKs in cancer and provide a mechanism for the RAF inhibitor resistance in cancer patients. Next, we will discuss a paper published in Cell on pain receptor and aging. Why this paper is so important? Aging and longevity has been studied for decades, but all players and mechanism are not clearly understood. Authors demonstrate a role for pain receptors in controlling aging. They also provide mechanism by which aging could be controlled. TRPV1 pain receptor knockout mice were used for the study. Longevity in both genders was extended to a similar extent with 11.9% increase in male TRPV1 mutants and 15.9% increase in median female lifespan compared to wild type. Also, prevalence of cancer tended to be reduced in TRPV mutants versus control mice. Long-lived TRPV1 mutant mice did not have reduced growth rates or altered IGF-1 levels, data not shown here. TRPV1 mutant animals retained a youthful metabolic program compared to age-matched control mice. In particular, the respiratory exchange ratio RER shown here, which measures the daily transition from glucose to lipid metabolism, displayed identical circadian oscillations at 3 months between both wild type and TRPV1 mutant mice from night to day. However, at late age, the TRPV1 mutants retained circadian transitions of similar order of magnitude compared to that of young mice, whereas wild-type animals lost their circadian shift. To uncover the mechanism underlying the youthful metabolic phenotype of the TRPV1 mutant mice, authors characterized the glucose metabolic profile of these mice. TRPV1 mutant mice were markedly more glucose tolerant than wild type at 3 months and 22 months of age, which correspond respectively to young and old time points in a glucose tolerance test. Fasting glucose levels were unchanged between mutant and wild type mice. Despite improved glucose tolerance, authors found that blood glucose levels from young TRPV1 mutant mice decreased by 40% following acute insulin challenge, contrary to the 97% drop in wild-type controls. Both old wild-type and TRPV1 mutant mice displayed age-onset insulin resistance, expressed by a diminished glucose response to insulin challenge relatively to a young time point. No difference in islet architecture when staining for insulin and glucagon in islets from mutant or wild-type mice, but increased beta cell mass was found in TRPV1 knockout mice. Quantitative RT-PCR analysis of pancreatic islet mRNA revealed that the insulin gene 2 was twofold upregulated in the TRPV1 mutant cells. Glucose-stimulated insulin secretion experiment GSIS revealed that insulin release was significantly more pronounced in TRPV1 mutant mice than wild type, thus explaining the improved glucose tolerance of TRPV1 mutants. Therefore, the TRPV1 mutant mice are not hyperinsulinemic but rather display an elevation in prandial insulin to respond more robustly to glucose challenge. To understand the mechanism, authors initially used C. elegans and later mouse model to establish the results. Results are summarized in a model here. TRP1 activation results in calcium influx and activation of calcineurin, allowing dephosphorylation of Krebs-regulated transcription coactivator CRTC1 and release from 14-3-3 proteins, resulting in nuclear internalization of CRTC1 and transcription of its targets such as CGRP and inhibition of insulin release. CGRP accumulation has a detrimental effect on energy expenditure, glucose tolerance, and aging. In contrast, loss of TRP1 promotes lifespan extension through increased insulin secretion, metabolic health by activating CRTC1 Krebs signaling cascade. This study provides a mechanism through which pain receptors are involved in regulation of longevity. Thank you for watching Breakthroughs from Science Mission.